My name is Dirk Voelkel. I'm the VP of Innovation for Cytiva, and I will be co-presenting with Ida Marie and Joel on their startups, but also on the Testa Center. So welcome to the session. I hope that I get through. I had some computer crashes before this morning, so I hope that everything goes well throughout the session. So first of all, Cytiva, you might have not heard the name. But you probably have heard from Pharmacia, Amersham, or GE Healthcare Life Sciences. We are a new standalone operating company under the Danaher Corporation, but it's the same as we were before. We have a new name. We were sold by GE to Danaher, but still the same facilities in Uppsala as a big facility and a big hub for us. So where does the name come from? Cy comes from cyto in Greek, which means cell, and eva is an ending from Latin that means doing and being capable of and something that you can do. So we're a global provider of technologies and service for developing therapeutics. And this is our mission and vision. So our vision, we want to change the world by providing life-changing therapies and make them available across the world to transform human health. And we do that by advancing and accelerating therapeutics. We're not developing therapeutics ourselves, but our customers do. We have all the tools and technologies that they require to either discover, develop, or manufacture drugs. So that's what we do. We're a very global business across the world, with many different hubs, but one of our biggest side, and that's why we're here also, is, I'm sorry, I'm just navigating here the platform a little bit. We're a global business, and we have different business units. Sorry, I'm just getting confused here on the platform. So we have a broad business ranging from research, as I mentioned. We have instruments and consumables for lab discovery. We are very, very strong in bioprocessing. So you can buy single individual skids from us, but you can also get a whole factory, if you'd like, for producing monoclonal antibodies or vaccines. And we work in cell and gene therapies, really life-changing therapies for people with different cancers that really rescue their lives. And you hear a lot more about the different technologies that we provide and where the future is from our CEO. So I definitely recommend you to go to the session at 11:40 by Emmanuel Ligner, The Future of Technology Within Life Sciences. As I mentioned, Cytiva is global. We're very far distributed across the world. We are where our customers are. We are where our patients are. We are where our instruments and our solutions are being needed. And as you can imagine, we need to be on the front end of innovation here. With our 7,000 people across the world in 40 different countries, we operate in many different locations. But we have 4 key hubs, and I mentioned Uppsala already. And in these key hubs and across the world, we have what we call innovation centers. Those are centers where we either directly or indirectly collaborate and train our customers and researchers across the world. And one of those centers is in Uppsala. And I'm very happy to share with you a few pages on Cytiva. On Testa Center. So Testa Center is a center that we have in Uppsala. It's a joint venture of the Swedish government together with Cytiva, which is a test bed for biology and technology, where we work with mostly startups, but also universities to verify whether they have an innovation that actually works. Very often they have something that works on a small scale. So we help them to get faster in actually getting to a larger scale, so you can industrialize their solutions. And we want to do that in an excellent way, of course. This is drugs, so we need to be safe and secure in providing the solutions. Let's go into a little bit more detail. And I think it's important to know that Testa Center is actually a separate organization from Cytiva. While we work closely with them, When you are coming in as a startup, your ideas and your IP doesn't get exposed to Cytiva. It's an independent company, it's a non-profit, and they provide lab space for biological production. Pre-GMP, so it's not for final production, but it's a space where you can scale up and rapidly work on unique equipment. They have really state-of-the-art equipment. You get access to expertise and services that is very hard to get to in a normal way. And they have a network of people across the industries. So it's a very nice way to actually scale up. What you get, or what the startups get out, is a risk reduction, a process know-how and scale-up know-how, and increased value for the startup company. And obviously, we're interested in doing this from a Cytiva angle because it allows us to really have the finger on the pulse on innovation, what our customers want, where we go, and we collaborate with a number of startups. So I'm very happy actually to introduce to you 2 startups that we collaborate with in Testa Center. And I want to introduce the first speaker here, which is Ida Marie Zintorn from Veronova. And Ida is the Chief Technology Officer, and she has been working with Veronova for 12 years. Coming out of the Uppsala University, she holds a degree in molecular biotechnology and worked in her PhD on computerized image analysis. So she took that knowledge and really went into and started a company called Veronova. And Ida Marie, over to you. And I'm very excited to have you on the session and share with you this, that you share with us the story around Veronova.
Thank you so much for that introduction. Can you see now?
Yes.
Yes, good. So I'm the CTO of Veronova. I have been there since before the beginning. So in this presentation, I want to share with you a bit about what we do, because I don't think everyone knows, but also more about our story with a bit of a personal touch. So my presentation will not really look like a, what can I say, professional company presentation, and how we got to where we are today. So we are a Swedish company, and we do electron microscopy combined with image analysis, and we do that for nanoparticle characterization. So we have a suite of products that we offer. We offer service where we receive samples from our customers. We do the imaging, we do the analysis, we send back a report. We also have developed software for doing image analysis and for getting quantitative measures out of what you see in the images of your sample. We have partnered with Hitachi to put our software in their transmission electron microscopy system in order to provide a kind of integrated full solution for automated imaging and analysis suitable for nanoparticles and specifically the pharma industry. Along the way, we also questioned a bit the giants in our field. Why does everything have to be so big and complex? And therefore to develop our own instrument. And out of that came Mini-TEM for miniaturized TEM. We did that jointly with a tech company in Czech Republic. And it's much more a conventional high voltage TEM. And it's also designed for easy automation. So a lot of things are easier to automate in the Mini-TEM than they are in conventional electron microscopes. I'll show you a little bit more about what we actually do actually. So our technology analyzing nanoparticles can be applied to very many different application areas. We, and I'll get back to we as a company, but we who were there from the beginning, we are all biologists or kind of biotech people from the beginning. So it was now into the bio area and specifically the pharma industry. So the focus currently is really what you see on top: vaccines, gene therapy, drug delivery, particles, and biologics. So what you see on the bottom is kind of what we want to explore in the future: pathology use as well as material science, which is a huge market when it comes to nanoparticles, where we have the domain knowledge. So that's kind of harder to get into. And then also other applications. What else can we use our software for? Can we put it on different machines and solve other problems and so on? So just briefly, you see to the right is Mohammed, and another person who's been there from the beginning, Josefina. She's head of the service unit. It was founded in 2005. Mohammed and I collaborated before that. I'm an image analyst, and he's a virologist. And he set out to found this company based on what we did. He was tired of looking at images, and he was tired of counting things and trying to explain the differences between viral appearance. So this is really a spin-off from the Karolinska Institute, where he was then. We are now more than 120 employees. And the next bullet, I'm really happy to say that we now can say that we have our headquarters in Stockholm. We actually do have offices elsewhere also, in Boston and then in London currently, but that's also something that we're expanding on. So our current main market, like I just said, is the pharma industry, and therefore I added these 3 images at the bottom. So these are electron microscopy images of adeno-associated virus samples, and it shows images from 3 different purification steps in production. So with other indirect methods, not imaging, it's very hard to assess what you really have in your sample. But with the image, you kind of see what you have. And the sooner you see and understand what you have in your sample, the earlier in the development process, you can kind of adjust the process in order to get high and good quality. So this is kind of a stepwise. To the left, you see less pure sample than in the middle, and to the right you see a really nice sample. And even so, once you have a really nice production setup, it's still worth to check the quality to see how many of them are intact and how many of them are broken and so on. So here you see a lot of really nice looking particles, but you also see some ring-like, and they are not intact, so they are kind of faulty particles. I want to say a bit about our journey, because this is a startup conference. So it was founded in 2005, and now it's already 2020. So I'm not sure you would really call us a startup anymore. But anyhow, so we kind of started. I joined, actually, I got my PhD 2005, and I left for a while to see what I knew in the real world, outside academia and outside the collaboration with Mohammed I already had. And then I came back late 2007. So that's when I joined Veranova for real. So before that, we had a small piece of software had been developed to do some particle counting. And then we kind of realized this is really hard to pitch. We talked with all the Hitachi and Thermo Fisher and Geol and so on, but it's kind of difficult to see how is this going to be useful. And then we realized, okay, we have a piece of software, but we don't really know what pharma is needing or how it should be used. So we started the service, and that was really to collect information and learn what does the industry need and how can we provide that. I think that was one of the best decisions we've made. And then we also realized that, okay, there are 3 giants in this field. There are 3 manufacturers of transmission electron microscopy, which are the machines that give the resolution necessary to look at particles at this size. We found another little company in Czech Republic who wanted to miniaturize things, and we teamed up with them, applied for European money, and 2011 we got a grant to develop the Minitem. It was super hard work, we actually managed, because out came the Minitem. So at about the same time as the Minitem was ready in the first version, 2004, our service had also grown. So there wasn't really a market for this because no one else had offered what we did. So already in 2014, we had 6 out of the 10 largest pharma companies as our customers. And then we realized that we need to kind of look ahead and try to grow in a better way, because we needed more staff and the demand for what we were doing was really high. So therefore, we also set up to try to start to acquire other small companies, electronics and technical consultants, in order to get more knowledge that we didn't have, but also to kind of get more staff. And if you hire a whole team, that's often easier than having to pick people one and one. If you have a good working team, they can kind of join easier. That's our what we've experienced. After the actual prototype of Minitem was ready, there's a lot of regulatory work and so on. So it was launched and we started to sell it 2017. And about the same time, we started to build our own service electron microscopy facility for GMP services. And the Minitem was marked in CE marked in 2018. And since then, we've tried to sell the Minitem. We have done that with some success. It's a new tool on a new market, so it's not easy, but it's picking up speed. So earlier today, we got our GMP certification, so now we're the only EM lab that is GMP certified in the world. We also continued to acquire tech companies in order to expand on what we need when it comes to instrumentation, camera knowledge, data analysis, and so on. And we also launched a new instrument together with Hitachi through a partnership. So briefly what we do with our software, the analysis software we do, you have it all in one. It's really important to have traceability in the pharma industry. So what we offer is really that you have all information in one place. So it's really easy to compare and to quantify what your samples look like. So we get the samples sent to us, we prepare them, we image them. The images goes directly from the microscope into the software. You run the software according to operating procedures, as everything in this world, and out comes a standardized report that is sent back to the customer together with images. The Minitem is another key for Veranova. That's why I talk about it. So it's compact design. It's really easy to use. So we designed it to be like any app or any software in your mobile or tablet or whatever. It has high resolution, not as high as the conventional electron microscopes, but high enough for what we mostly try to do. The analysis software is integrated, so you don't need to move between softwares for imaging and analyzing things. You can put it in any lab. It doesn't require anything more than a normal power outlet. So far, to come to where we are today, our strategy from the very beginning was, of course, you need money. So our strategy was to go for research money. And I think that was pretty wise, because in our customer field, it's all academics, and you need credibility, and that you get through these collaborative projects. You publish, and you also get access to cutting edge knowledge from academia and partners. Another strategy was to set up the services. We realized that we need to learn what the industry wants and what the industry needs, and how, in order to design how to provide that. And I think if I compare what we do with the other electron microscopy giants like Geol and Hitachi, and Thermo Fisher, we have actually managed through our service and through our credibility to get into the pharma market, which they have not. So they have, through our whole journey, been extremely curious about what we do and how we do it. We say no to no one, so we also have partnerships with both Hitachi and Thermo Fisher, actually. And early on, we also established a close collaboration with DeLong Instruments, who provide the hardware for the Minitem. You can't be best at everything, so you better pick your partners that are the best in what they do. And then more recently, now that we're beginning to kind of pick up speed, we have the strategy of acquiring additional expertise and knowledge, and also kind of ready and good working teams. So in order to succeed, which I think we have, I still think we can grow a lot, but I see it as a successful company. I think you really must believe in what you do. It's a struggle from beginning and still is. But you need to have a core of some technology that is useful for someone, and you really must believe in what you do. We created a market that didn't exist because we thought that this information is really valuable, and why haven't anyone made use of it? We made it. We now have a queue for our services, which is extremely we're so happy about that. We've also throughout the years been very kind of careful when selecting new colleagues. From the beginning, you're a small team. You know everyone and you know everything, so it works smoothly. But as you grow, that becomes more difficult. You also need more different profiles and different kinds of people. But we have a very careful hiring strategy. You need to be very persistent, and you need to be dedicated to what you do. If no one is there, if you don't have anyone for a specific task, everyone has to kind of pitch in and do it. And based on our story, I would also like to say, dare to do differently. We challenge the giants. Why are the instruments so manual to run? Why are they so big and clumsy? And why does it take forever and no automation to get to these images and the results? So that's how we got to where we are. And now what? We're part of
that. Just can you wrap up your kind of on your time?
Yes. Sorry for that. So we grow where we stand. Testa Center is important for that. We've gotten the opportunity to put Minitem there for the Testa Center clients to try out. We get knowledge, what they need, and they get to see our instrument, which also serves their purpose. We, of course, want to automate more, and we also want to put our software on more machines. Thank you.
Well, thank you very much. It's a very exciting story, and I love how you actually learned your way into it. It's something that we're actually also propagating and doing internally as well, where we try to encourage to have a learning environment where you go in and you learn from and with your customers. Mm-hmm. I have several questions to you, but I'll hold them till the end, and I would like everyone else to do the same. Please just let us know questions that you have in the chat. But I want to give the floor to Joel. So Joel joins us from SCIAR. He's the CEO of SCIAR. He studied physics like I did many years ago, but he pivoted actually to studying molecular biology, which I didn't do, but I actually work a lot with molecular biologists these days. So it's very exciting. And I think he met with a co-founder who's a pilot who did a lot of work on human-machine interfaces, and they figured out that actually the current lab environment just is not leveraged enough, and they have some ideas on how to improve it. So Joel, very excited to have you here. So please share your story of SCIAR.
All right, thanks, Dirk, and thanks everyone who is participating here. So yeah, my name is Joel. I'm the CEO of SCIAR, and Dirk gave my short kind of CV there. So let's get down to it. So not sure about what is people's background, so I just kind of frame this a little bit. So what we have is a software tool for scientists and lab technicians to make their work easier and automate documentation and get rid of human errors. And here we have kind of outlined the basic process for pharmaceutical industry, but you could do this with a lot of different kind of industries, like in life science, biotechnology, wherever. So what we have is on the left side is basically the academic basic research, industry R&D in companies and other organizations who are compiling this, accumulating this data for the actual research that then we can make our way to new drug discovery and doing preclinical studies, where we then go to the clinical phase, which lasts as long as the at least as long as the first part. So we focus on the left side where scientists and lab technicians are actually doing the lab work and studying the molecule, studying the biology to understand what makes us tick and what we can leverage to use to make new therapies and new drugs and new treatments. But the problem is that this preclinical phase is funded. It's about 56 billion each year that US in USA you put into this this preclinical studies, and it's about half of that, 28 billion each year, that are kind of wasted for funding experiments that cannot be reproduced. This was a study published a few years ago, and it showed that, well, this is of course an estimation, but we see about 70% failure rate when we're trying to replicate other people's scientific experiments. And even though we try to replicate our own work, we still see about 50% failure rate. This is something that wouldn't be acceptable in any other industry. And as I have worked in a few labs, I understand that this happens, but something needs to be done. So yeah, so what is the reason for this? Well, we point fingers at study design, faulty reagents, or poor laboratory protocols. But what we see that this comes down to is basically just poor quality data. Like we don't have good evidence chain from the moment the reagent was manufactured. What happened to it? What pipette did you use? What lot number was that reagent you used? This is something that just there isn't resources to do this, and there hasn't been any good tools for this for so long, and that's why a lot of data is still gathered in even bigger companies in life sciences. It's still gathered with the pen and paper. But like I said, until recently, there hasn't been a good tool for this, because this is what our company is working on. So what we have is we have this cloud software, which is basically a very smart electronic lab notebook, but it's also kind of brain for the whole lab. So that's where you have your, you can design your protocols, you can see your documentation, you can share your experiment documentation easily to other users, so they can replicate very easily your experiments. If you're an administrative person in a lab, you might be interested to see where are your different projects, what resources do you have, what do you have to maybe order more, these kind of things. And what we use in addition to this cloud software is the AR, so Augmented Reality. And that's the glasses you see there. We use the Vuzix AR glasses, which are very lightweight and they're comfortable to use for a longer period of time. And what it does, it shows you the protocol step by step. And it's very easy to follow it, and it's very hard to make a mistake, because we also use the camera to identify certain things. Like, let's say you have to identify the reagent you are using. So you are just reading the QR code, which is there by the manufacturer, and we get exactly the data. Which reagent was it? Was it the correct one? Which lot? And so on. We can also get more data about how many times it has been used and when it shows some signs that it might be a good time to open a new bottle and so on. So this is our product in nutshell. So it's a kind of new way to boost laboratory work. And one thing we are also kind of planning as well is to add more automation. So our platform allows also easier way to add lab automation, because you don't want your laboratory kind of work go from documenting excessively to programming automated systems excessively. So our kind of user interface allows this easy workflow when adding automation. So a little bit about our history. Yeah, like Dirk said, I met my co-founders. Well, one co-founder was my classmate who was previously also he was a airline pilot, and he has been working for a lot how to make human work standard in those airplanes. So and my other co-founder I met at a lab I was working at. So then we just started talking about the possibilities of AR at some point. We just kind of got exposed to it, and we just put two and two together and thought, well, this is something we should definitely do, or at least look more into. After some initial research, we found out that this actually looks good, and we founded the company in 2018. Pretty soon after that, we in Slush 2018 in Helsinki, we met Jens Hammarlund from Testa Center, and he was kind enough to invite us there to get to know the Testa Center very well. And in May 2019, we were actually able to conduct our first pilot or test in Testa Center with some Cytiva employees and some students from Uppsala University. And this was, I would say, this was very big kind of step for us at that point, because we were just beginning, and it was kind of very important to us to get actually real life evidence. Like, are we on the right track? Is this really helping? Is there something we haven't thought of? And this is kind of where the value from Testa Center came from. That we were able to go there, and that also gave us a little bit of a name. Like even in that stage when we were just a young company with like 6 months of track record of anything. So yeah, and we were also Testa Center's first digital project, and also their first project from abroad. So yeah, the next big thing for us was our first kind of actual pilot with the University of Helsinki and their Faculty of Pharmacy. This is there we provided the AR software for the faculty so they could make a case study how AR can help in laboratory teaching. So it was kind of the same product that we are developing now, but it has some. We had to kind of optimize it for teaching, because it's a little bit different kind of scenario when you're trying to teach people instead of just trying to catch every error that we try to do. But that gave us pretty good results, and now we are also going into. We can go into education as well. So, what does the future hold for us? Well, 2020 was, of course, like for anybody, not optimal because of the coronavirus, but we were able to get some Business Finland funding to get us over the situation, and we were able also to conduct some multiple pilots during the summer. So now we have been doing our product development and business development, We are actually planning to launch our product by the end of this year, and hopefully next year we will be expanding first through Nordic and Baltic region, and then to the rest of the EU, and hopefully to the US markets as well. The companies listed there are companies we are right now negotiating our pilots with for this year or beginning of next year, and we have some others as well in the pipeline. So this year looks actually pretty interesting to us. This year we are also looking for additional investment. We are hoping to close this round by the end of the year. So let's see how that goes. Anyway, I think that's pretty much all I have for now. But please take my contact info. You can always either contact me directly if you want to hear more about the our investment opportunities, or if you have a company that you think might benefit from our products, just contact me or go to our website. You can there book a demo directly. So thank you.
Thank you, Joel. Exciting story. I really didn't know that 50% even of reproducing the own work didn't work. So I think there's a lot of way to go and really avoid the waste. I think it's a great story. And I love the story that you've gone through. So we got some first questions coming in. Again, everyone, if you have questions, please leave them on the chat. So maybe the first question to Ida. Ida, what has been the limiting factor for growth, and what are the biggest challenges that you're working on right now?
I think common for all startups is money. But having been, we've been around for quite some time. So I would say, looking back, it's money from our side and it's money from the customer's side also. I think the challenge is we went into a difficult business. It's difficult technology, and it's a difficult market because it's so very regulated. So coming in with a new technology and getting the acceptance is a challenge. So I think our starting approach to collaborate to get credibility and also to start the service part to get acceptance for what we do has really been the key for us. It's still the challenge. The regulatory side is huge and growing, and the more you offer, the more it needs to be compliant to everything also. So now that we have the acceptance from the pharma industry and they want to move it into production and so on, there are new regulations and markings and certifications that you need and so on. So it is a difficult market. So of course you need more money, but I also think that you need time.
Yeah. Well, thank you. This is great. The regulations is something, particularly in our industry, right, that you need to work through. You have something that works in the lab, but once you want to scale it, you need to be compliant more and more, and particularly if you use it in the drug development process. Yeah. No, agree. Joel, a question to you. Like these headsets, I'm wearing glasses, right? And if I go to a lab, I have to put safety glasses on top. And do I have to use AR on top of that, or how does that work?
Yeah, good question. And that was actually something we were kind of worried about 2 years ago. But since then, the technology has come pretty far. And let me just quickly change to that slide. These glasses that you see here, they don't have any lenses, and you can use your own glasses or safety glasses. And these are, we use actually in labs, these headbands, which kind of allow you to just have it kind of, it doesn't really affect the use of the glasses. It's kind of external on your kind of field of vision here. But it doesn't, especially with the headband, it doesn't affect the use of glasses. It doesn't weigh practically anything. So it's kind of just as if you had a separate display floating around here. So that's how we have been able to kind of tackle that possible issue.
Great, thank you. There's one more question, maybe actually maybe to both of you. Like you're both entering a space that's regulated, and Jesper asked a question here. If you had a wish free, so genie out of the bottle, and you have one wish, could you see a different way of working in the regulated space without compromising compliance? So I think it's an interesting open question here. Any ideas?
You can go first.
Well, thank you. I find this question interesting, but also very challenging. I mean, I do understand why you need the way it works now, but it's difficult when you are kind of at the cutting edge. So we work a lot with AI and new technology, and the IT industry and the tech industry is developing tremendously fast, and it feels as if the regulatory world kind of applies to what was yesterday. And to us, that's extremely frustrating, that how can we fit the new and much safer way of working into this regulatory world, because it feels as if they are also not using the best working ways, because it's kind of made for the technology that existed yesterday. And I think that's very frustrating. And we are discussing with the FDA, for example, and I know that they have their working groups and so on for how can we fit new technology into the system in a good way. But I don't have a solution, and I don't know how I would do it. But I would also say that it's been still is a heavy burden on us, and it's kind of a lot of our resources go not to what we think is maybe best for the customer, but rather fulfilling the regulatory work.
Yeah, I would say of course, I would say for us, it's maybe actually, I would kind of say it's not even that much of a problem maybe for us. Of course, we have to also fill the regulatory kind of milestones for GMP, GLP, or GXP, or whatever you want to call it right now. But we have been looking at, let's say, the new EMA's guidelines for data integrity, and we kind of see that it's more of our clients that have to fill that regulatory compliance, and the way they can do it is through tools like ours. So I'm not sure if I would want to change too much about it, actually.
Okay. Yeah, and I think, I mean, we have examples where, I mean, with our BioCore platform, for example, many years ago, 30 years ago or so, when that was started, it was also a new technology. But once, Ida and Joel, I think once you establish yourself, if you're then a recommended stage in the regulatory process, then it also gives you a nice protection in a way. So there's a double-sided sword a little bit to it. But I agree that getting in, and I have way more questions. I would love, for example, to ask you how COVID accelerates what you're doing or slows you down. But I think the session here is over, and it will. We're starting to lose some people here as well. What I would like to do and invite Joel, Ida, and whoever is interested, please join us in the exhibitor booth for Cytiva, and we can then have an open chat there as well. That should work, and then we can have more questions asked there. So thanks a lot. A great session, Ida, Joel. I thank you very much. I want to also to UPPSTART to make this possible. I'm so happy that we can actually meet in a conference, even though we can't meet in person. It's a good interim solution here, I guess, until we can meet in person again. So thank you very much.